Okay, so we're gonna start off today by finishing the last three whole slides that we had for chapter two, and then we're gonna start out on chapter three. Um, so if you've noticed, I didn't post all of the chapter three slides yet. Mostly I wanted to see how content happened today to know how much I want to cover. It's one of those chapters we can go into as much or as little detail as is necessary. But luckily, we have lots of time. We have lecture and Friday, as well as all next week. So we have lots of time to make sure that we get all the content we need to. But for finishing up with ethics, you might already have a general idea of what ethics are, but they're basically a set of principles that describe how people should be educated, treated, and respected when they participate in a study. So this is a definition that's pretty biased towards psychology participants, so specifically towards humans and humans who are doing psychology experiments, but that kind of makes sense for the course that we're in. I do always make the disclaimer that there is an equally as intense set of ethics that apply for non-human subjects. So that's actually what I'm a lot more familiar with because I've done a lot of work with birds. So same basic idea, but with the wording changed so that we're doing these principles as they apply to non-human animals. So just keep in mind that these are human-centric. Too far. So our general principles of ethics tend to be divided up into more or less five categories. So we have five principles here. You might see them condensed down into three, but even when they do that, those three principles include the same five as sort of subcategories. So we'll stick with the five. So the first of these, principle A, is called beneficence and non-malfeasance, or yes, we'll call it good enough. Basically, what they mean there is that you should be treating people in ways that benefit them, don't cause them undue harm, and ensure that your research is something that will benefit society. If you have to make people uncomfortable, if it's going to be sort of not the best of situations, you have to show that this research is worth doing and that the benefit to society as a whole is sort of more important than a little bit of discomfort on a small scale. So this is where our ethics committees come into play, where they'll make sort of evaluations of whether or not something qualifies, whether or not it would have a large enough impact or potential benefit to society to make it worth conducting. Our second principle, principle B, is fidelity and responsibility. So basically, we want to be trustworthy, behave responsibly, don't lie, don't sort of mislead people, don't do bad things, be responsible in what you're doing, and take ownership of what you're doing. So, pretty straightforward. Our third principle is integrity. And so here you want to make an effort to be accurate, truthful, and honest in your work. Basically, as a scientist, you want to make sure that you are doing the best science that you can and that you're not doing things like falsifying data or misleading participants or intentionally introducing bias in order to get a better result or anything like this. So it's basically saying have integrity, be a good scientist, follow the rules, that sort of thing. Our fourth principle is justice, and this is a huge one. This is basically saying treat all groups of people fairly. Make sure that when you're selecting samples, that you select equally from all of the populations that will benefit from that research, not just those who are, say, convenient or easy to manipulate into participating, and be aware of biases that can exist. This is sort of a big one, and it's one of the major reasons that kind of drove ethics into being such an intensely regulated thing in the scientific field. If you've heard of it before, so there are, well, actually, there's many, many examples we could draw from. A couple of ones off the top of my head are things like the Stanford prison experiment or the Tuskegee syphilis study. Those are things that happened before ethics boards existed, and they're things where people were unjustly treated. If we look specifically at the Tuskegee syphilis study, um, we have a very biased sample where basically they targeted primarily low income Black communities and didn't treat people for syphilis, or even in some cases gave them syphilis to see how it would progress. Now, the way that we apply it to this principle, that's not an equally selected sample. They went after a marginalized group, and the benefits of that study, anything they had learned from it, could benefit all humans, but they targeted one specific group. So that's one of the driving forces that pushed all ethical boards and all of these ethical principles into place. But it's very applicable to this specific principle. So I mention it there because I teach another class. It's an entire semester on psychology research methods, and we have like entire chapters on ethics. So we could spend way more time on this, but just making you aware of the things that have occurred that have sort of led us to where we are now, and why following these principles, why keeping these things in mind is so important for research going forward. And then our last principle here, principle E, is respect for people's rights and dignity. So you should be able to recognize that people have autonomy, respect their rights, ensure that they give informed consent, and take any necessary precautions to avoid coercion. And we'll actually talk quite a bit more about um, informed consent, drew a blank for a moment there, and coercion in just a moment here when we start talking about sort of special considerations and populations that may not be typical populations. So all of this is basically saying that they have very strict guidelines that state what you can and cannot do in research. There are internal review boards, or IRBs, that will review and assess all of the studies that are conducted under that particular institution. So if there is research conducted on a university campus, that university has some kind of internal review board that will oversee approval for all studies that happen there. And this is something that's sort of closely monitored. There's strict penalties if you violate it. You can actually end up blacklisted from conducting research, even in the entire country. So it's a pretty intense thing if you violate these principles. So just they're not just something that's put on paper, it's something that's actively monitored and enforced. All right, so onto our special ethical considerations. We have to keep in mind that not everyone is able to give informed consent. So we've mentioned this term a couple of times, but if we break down what it means, consent is that you are saying yes to participating in a study. When we say that it's specifically informed consent, we're saying that you have to have a necessary, like all the necessary information to make an educated decision about whether you choose to participate or not. So, in order to give informed consent, you need to have a basic understanding of any potential risks as well as benefits from that study. So, you need to make a choice for yourself. But you can probably think of some groups of people who can't give informed consent. One of the biggest ones to consider are children. Children might not have the capacity to understand what's happening. And so, if you wanted to do a study involving children, you can't get informed consent from the participants, but you'd have to get it done from their parents. So, their parents or guardians. And the same applies for, say, those who have mental disabilities. If they're not able to make informed decisions, then you would have to get consent from a parent or guardian. So someone always has to understand what the potential risks and downsides are, as well as what the potential benefits and upsides are. So that's one of those big things that are really important. Another thing that we might want to consider here that, again, I didn't really think of it too much until I taught my methods course, where we spent some time talking about populations that might feel coerced into participating in studies. So the example that they've used is prison populations. If you want to study, say, educational programs in prison infrastructure, You have to be very, very careful because that environment actually leads prisoners to think that they have to participate in a study. They might feel that if they say no, if they turn down an offer, they might be punished. It might go on their record, it might affect their release or anything like that. So we have to have very sort of explicit steps set up, and we have to make sure that no one feels like they need to participate if you're going to be using those populations. Because if somebody is feeling pushed to participate in a study, then that violates that whole principle and that goes around those ethical considerations. So it's something we have to be very aware of. It's another reason why there's some questionable things about, say, offering monetary incentives for people to participate in studies. This is sort of off the path of what we technically need to cover, but I found it interesting because if you offer a very large sum of money for people to participate in a risky study, then that's biasing it towards those who are lower income or not well off. So you're kind of biasing your selection process for the particular group, and then you're not having a representative sample of all who could benefit. So I guess that's one of the reasons they can point to for in Canada, you can't be paid to donate blood. And that's one of their ethical reasons why. But that was a tangent because it just occurred to me. Our last slide here is you might have made the connection already, but if we have to get informed consent, if we have to tell people about this study for them to participate, can that create problems with some of the stuff we talked about earlier? So if we tell people what our study is about, what if they change their behavior based on their new expectations? What if they adjust the way that they answer questions to match what the theory is? Or what if they're being jerks and they go the opposite direction? Um, so this is one of those things where we have to decide what is the best practice. So some studies might not reveal the true nature of the study until you've completed your participation. Anytime that you don't reveal everything, we call that deception. And what you'll typically see in a study, I use the example from one of the studies I actually helped set up. I don't really do human stuff. I know people who do, so I understand how they work. But they were getting people to listen to Black-capped chickadee songs, and the songs were drawn from two different populations. Some were from Ontario, some were from BC. And the point of the exercise was for people to sit and listen to these songs and see if they could pick out which ones were from Ontario and which ones were from BC. So they would click on a computer screen, which group does this sound belong to? Now, they didn't tell their participants that they were listening to Black Cap songs and that they differed based on location. So, what they said instead was that you're going to hear some sounds and you're going to put them into two categories. We'll give you feedback to tell you if you're right or wrong, but that's all you need to do. They also listed all of the risks associated with this kind of a study. So, you have headphones on, auditory exposure. If you're doing it for too long, it can be a bad thing. You're staring at a computer screen, that could be hard on the eyes. You're using a mouse to click, repetitive motion can be troublesome. That's the kind of detail that you go into for ethical things. Um, and then once they had completed the study, they did something called a briefing where they sat the participants down, give them a nice printed sheet of paper that says, for the study that you did, the sounds that you heard were chickadee songs, and they differed based on location. This is why we were studying it. Here's a little bit of a summary of our research. If you have any questions, here's our contact info. And so at the end, if you've had any kind of information that's not disclosed, you can disclose it at the end. But in order to do that kind of a study, we had to go and get approval from our internal review board that said that that amount of deception is fine. And because we still told them the important risks, it's okay to go ahead. It's still an ethical study. Sometimes you have to be a little bit more extreme. And in those cases, your review board would have to decide if the potential problems of excluding information are outweighed by the potential benefits. So that's why those sort of separate parties that aren't people who are running the research are in charge of making those decisions. But these are things that researchers have to always keep in mind when designing studies. Is this something that is ethical to do? Will we get approval to do it? And if not, how can we make it better and more in line with those principles? So that's the very, very short version of ethical considerations as they apply to humans. As I said, we could spend hours and hours on this, but we'll leave it with just those three. We are, however, so chapter 3 is where we start talking about the biological basis of behavior. Before getting into this chapter, I always kind of give people a heads up that we've sort of been building up information. We started with what is psychology in general, how does psychology work, what are some of the important principles we want to keep in mind. Then we moved into what's science in general, how do we do science, how do we think about science. And now we're actually getting into some of the parts of the body, some of the processes that are going to be directly related to behavior. So again, we're building up information, but I find that this is the chapter where we finally start getting into really like real content, as it were. So this is a dense chapter. That's why I'm not too set about being ahead to get started on this. It gives us some time to slow down and go over stuff a couple of times. And again, I give that as a disclaimer because I won't just go over some of these topics once and then hope you've got it and move on, because a lot of these are a little bit more complex than anything we've covered so far. So what I like doing is ending right after something's been explained, giving you some time to mull it over, and then coming back to the next class and reviewing it again, maybe with a little bit of a different perspective, or maybe with a video or something like that. That kind of repetition gives us the chance to sort of have it sink in a little bit more, but it also gives us the chance to correct any misconceptions the second time through. So just a heads up, it's going to be a lot. Um, if you've already done a cell bio class, then you already know all of this, and that's fine. But it's a very interesting topic, and hopefully it's presented in a way that's kind of interesting too. So with all of that, hopefully I haven't scared you all off. But let's start by talking about sort of our smallest component, our building block that we care about for our nervous system. So we're going to look at the level of the neuron. So neurons are cells. So a single neuron is a single cell. And these are specialized cells that can convey sensory information from the environment into our brain. They can carry out operations within our nervous system. They can allow us to make associations between things. And they can transmit commands to the rest of our body. So we can send signals from our brain to our muscles in order to carry out actions, all using neurons. So neurons are very specialized and have lots of different functions. And we'll talk about sort of three broad categories of them that kind of encapsulate each of those three basic functions. And so you've probably been introduced to a little bit of cell biology, probably in terms of even high school bio would talk about this is a cell and this is the cell's nucleus and things like that. We're going to go more in depth into some stuff, and we're going to go much less in depth into other stuff, depending on how relevant it is. So let's start by hopping into our stereotypical image of a cell. We'll also kind of deeper a little bit later and look at some of the other shapes and forms they can take, but this is typically what people think of when they envision a cell. And we're going to point out some of the important components here. So the first thing that we care about is the thing right in the middle, that sort of big yellow spot in the center. That's the cell body, also called the soma, S-O-M-A, in case you care. And that's basically where all of the stuff that a normal cell needs to function is going to live. So we can see they've labeled the nucleus. The nucleus contains all the genetic material that the cell needs to function. It's where all the organelles would be. So that's where the mitochondria is doing its energy things, and the ribosomes are doing protein manufacturing things. We sort of hand wave all of that and say that in the cell body, this cell is being a cell. And for the rest of it, that's where we're looking at specialized functions that are specific to neurons. So the next thing we look at are our dendrites. So these are the little branching things that stick off of our cell body. And the dendrites, you can think of them as sort of antennas that are interpreting or routing incoming signals from other cells. So the dendrites can receive signals from other nope, not nucleus. I'm looking at nucleus, other neurons. And so they can input information from there. They can also receive information from other sort of sensory receptors. So receptors in our skin would be tactile receptors, and they can send a signal through the dendrites and into the rest of this neuron. Um, we have in the middle a couple of terms that we won't care about right now. So things like the axon, helic, and the myelin sheath. We'll get into those in a little while when their function becomes relevant. For now, what we care about is that long, sort of sticking out bit that's wrapped in the myelin sheath, and that's our axon. The axon is a long, thin protrusion from the cell body, and if once we start talking about signals moving from the cell, signals are always going to be moving in the same direction. So the signal comes in at our dendrites, so that's those antennas putting stuff in. It's going to pass through the dendrites, the cell body, and then out the axon. The axon is sending that signal in the direction of those red arrows towards the end. And the end of our axon is called a whole bunch of different things. I like to call it the axon terminals. You'll sometimes see them just called terminals. You'll sometimes see them called axon buds. Um, Everybody has a different name for it. So just giving you that acknowledgement that others exist, I will typically call them axon terminals or terminals. But those are basically like little protrusions out of the end of our axon, and they end in little bulbs. Those are the terminals, the bulbs, that increase surface area and allow good communication from the end of this neuron to the dendrites of the next. So that's where that communication is going to pass on to the next cell. And so we're going to talk more in detail about what specifically is happening in between, but this is meant to sort of ground us and give us an idea of what we're going to be looking at. Okay, so now I have those three types of neurons. So the first of these are motor neurons. And this one here is actually a motor neuron because we see that it's connecting to a muscle fiber. So it's sending a signal from the brain, from our central nervous system, out to our muscles or our organs. So we'll talk about the organization of our nervous system, but our brain and our spinal column are the central nervous system where sort of that decision-making process is happening. So when we're talking about things coming in or going out, we're talking about them coming in or out of that central nervous system. So they're coming into the brain or leaving the brain, usually via the spinal cord. So our sensory neurons are the ones that are bringing in information from the environment. They carry information from the body and the outside world into the brain and spinal cord. So that's when I was mentioning our skin receptors. Those would signal our neurons and then tell the brain what they're feeling. If we're smelling something, if we're tasting something, all of that would be relayed through sensory neurons. And then in between our sort of input and output, that's where we have our interneurons. And they work in between everything else. And they basically make connections and allow us to process things. These interneurons are very diverse in functions. We basically call them the ones that are in between, and they make connections. And so this is more of a for your information kind of slide, as opposed to like memorize this. But I just wanted to give you an idea that even though that one on the far right is our sort of prototype of what a neuron should look like, Neurons can have lots of different shapes. So the one we have been looking at would actually be called specifically a multipolar motor neuron. So the motor neuron part we could probably get because it's connecting to a muscle fiber. It's multipolar because there are multiple or lots of those dendrites sticking out of the cell body. But for our variants, we can look here at the top, we have a unipolar sensory neuron. And so we know it's sensory because it's connecting to our skin, so it's those incoming receptors. And it's called unipolar because our dendrite, there's only one of them, and it connects directly to the axon, which is atypical. It bypasses our cell body in the middle. And the reason why it does that is actually to help transmit a signal much faster. So by having the cell body off to one side, our signal can go straight from the dendrites to the axon without having to push through that whole cell body. So that's a convenience thing. That's a speed of transmission thing. So they have that shape if they want to send a signal very quickly, usually over longer distances. We can also have bipolar sensory neurons where we have one dendrite and one axon, and they both come out of sort of opposite sides of the cell body. That's a little bit more typical for a sensory neuron, but again, different from our multipolar motor one because there's only the one main branch of dendrites. And the last thing we can talk about are the interneurons. And these ones break all the rules. We label one thing and it's the nucleus in the middle. They're sort of a mess of lots of dendrites, lots of stuff moving around, and they kind of change form based on function. So we don't talk too much about those. So we can still continue assuming sort of a motor neuron stereotype, but I just wanted to give you a heads up that there are other kinds and they can all have different functions. So there are other options. All right. So now we're going to get into the actual cell biology type stuff. So like I said, if you've recently taken cell bio, this should be a little bit of review. But you might recall that our cell membrane is made up of what's called a phospholipid bilayer. And so what that means is that there are two layers of this molecule, which is a phospholipid. And if we talk about the structure of the phospholipid, it actually explains why it works really well for the role that it's in. So if we think of these, so these are a bunch of kind of like little balls with two tails off the ends. The balls, we can call that the head of the molecule. And that's going to be our phosphate group. And so our phosphate head is actually hydrophilic. We look at the roots, hydro means water, and philic means loving. So the heads of these molecules really like being in water. It means that they'll dissolve in water when exposed to it, which is convenient because a lot of the fluid that surrounds our cells and that's in our cells is at least partially water. So those heads don't mind being surrounded by water. They'll happily face into either the intracellular fluid or the extracellular fluid. Intra meaning on the inside, and extra meaning it's external to. That was always one of those things that confused me. So that's the heads of it. But what about that lipid part of the phospholipid? And that's where those tails come in. So there are two lipid tails, they're fatty tails, and they are hydrophobic, so afraid of water. They want to be away from the extracellular fluid and the intracellular fluid. So we'll notice that all of the tails have kind of clumped together and they've set up these two layers where they can hide from all of the water and sort of keep it out. And so here we have our phospholipid bilayer. So we have the heads on the outside and the inside, and that'll dissolve really nicely with the water. And we have the tails in the middle, and they want nothing to do with the water. And that does a really good job of preventing anything from passing from the outside to the inside unless we allow it. Because if something were to pass across, it would have to have the right charge to be able to interact with things that can interact with water, but also interact with things that don't interact with water. and stuff doesn't do that well. So this is effectively something that you cannot pass through, which is convenient because then we can start looking at what happens when some things are inside, and some things are outside and they'd like to move. So we can introduce our next concept, which are protein channels. So again, different stuff with different names. Sometimes you'll see them called ion channels. Sometimes they're referred to by exactly what kind of molecules they allow to pass through them. So they're all the same concept. But here, these are the proteins that are inserted into that phospholipid bilayer. They cross the cell membrane and they allow specific ions or specific molecules to pass from outside to inside or inside to outside when they're open. And they can specifically be opened by different types of signals. Some of them are what we would call electrically gated, which means that they are switched on or off using electrical signals. And some of them are also, I'm drawing a blank there. Some of them are chemically gated, which means that specific chemicals or specific molecules, if they bind to it, that will turn it on or off. We have different ways of opening and closing these channels, and each of these channels is usually going to work with one specific kind of molecule. So we're going to talk about sodium ion channels that allow sodium ions into the cell, and we'll talk about potassium ion channels that allow potassium out of the cell. So that's usually how we refer to them. So whenever we talk about something something channels, that's what they're doing. Okay, so that's set us up for what kind of structures are going to be important. We're going to introduce one more structure that's really essential, but we're going to hold on to that for just a little while. And first, we're going to talk about what happens when we have different molecules inside versus outside of the cell. So the first term that's going to be important is the concept of polarization. And this is a state where there's a difference in electrical charge between the inside and the outside of our neuron. So the inside of the cell and the outside of the cell have differing electrical charges. So we can say that a membrane is polarized if the inside and the outside have different charges. We can describe the magnitude of the difference in charges using something called voltage. So voltage is specifically a measure of the difference in that electrical charge between 2 points. So you can't have like an absolute voltage. We can't just say what the voltage is inside. We have to say what the voltage is inside relative to the charge outside. So the voltage is giving us that comparison across the membrane. It's not giving us an actual voltage at a single point. So this is a comparing between. So when we talk about a voltage across our cell membrane, we're comparing the charge inside to the one outside. And that's why we're going to be looking at something relative to sort of those points. And then having a charge across our membrane can actually set us up with something called a potential. So if you've taken sort of, I guess we can see it in physics or even chemistry, when we talk about potential, we're usually talking about sort of stored potential energy. And that's basically saying that stuff is held in a position that it would like to move from. So when a neuron is at rest, there tends to be a charge across the cell membrane, and that charge would like to equalize, it would like to move around. So by holding a charge across the cell membrane, by not allowing things through our channels, we're sort of storing that energy like a battery, where once we flip the switch, once we open those channels, we can harness that energy, we can use it for something, but while we're sort of waiting, while the cell is doing nothing, we have what's called a resting potential. So we store a difference in charge between the inside and outside of that membrane when the cell is at rest. And so all of these terms, they might not quite make all sense right now, but we're going to be using them as we work through all these topics. And again, we're going to spend quite a while on this. All right, and then the last thing that we want to introduce are our ions, and then we'll talk a little bit about how they move and how they want to move. So ions are just atoms that have lost or gained electrons. So they are molecules that are either positively or negatively charged. We're going to look at a very simplified version of this. We're going to care about, for our positive ions, we're going to focus a lot on sodium. So Na plus, that's just a sodium atom that has lost an electron. Electrons are negatively charged, so if the negative goes away, then you are net positive. We also have potassium, which would be K plus, which is again just a potassium that's lost an electron. For our negative ions, we have chlorine, so it's listed as Cl minus, so it's a chlorine ion or a chlorine atom that has gained an electron. It has an extra negative charge. And then the last one, if you know any chemistry, you might be saying, hold on, there isn't an A element. That's actually just one of those weird biology notations, and that's meant to be an anion. And this is just, they use A minus to indicate a whole bunch of different negatively charged organic molecules. So anions are negative ions. So that's sort of a weird notation thing. So if you see A minuses floating around, that's them simplifying a bunch of different organic molecules that can be there. I just found it weird that they grouped it in with ions, but it has charge, so that's and then at the bottom, we have this figure that we're actually going to look at a couple of times and focus on different aspects. But for right now, what we care about is the fact that there are different ions outside than there are inside. On the outside, we have more reds. On the inside, we have more blues and greens. And so that's helping us set up that resting potential, that difference in charges across the cell membrane. And we can remember that our phospholipid bilayer is keeping them separate. They're not allowed to cross through. And in the middle, we have one of our protein ion channels. And if it's not open, it doesn't allow anything through either. So they're stuck being unhappy on one side or the other of the membrane. Okay, so next we're going to talk just a little bit about why these ions want to move. This might be a little bit more than what we need, but that's fine. I also apologize for using PowerPoint animations. They're like against the core of my being, but it is the best way to show this, so I've had to just suck it up. So the first thing we want to consider is the force of diffusion. So this is that idea that ions want to move away from other ions that are the same as it. So we have a bunch of potassium ions here, and they would like to move through the membrane so that they can spread out. There's sort of they have sort of forces between them. There's too much pressure on them. They would like to spread across. They would like to diffuse and spread to fill the entire space if they could. So this is saying that gray line being the membrane, this is what they want to do, not what they necessarily can do. So they're going to spread out. Some are going to go inside, some are going to stay outside, but they're going to take up that whole space that's available to them if they can. The second thing that can motivate them to move is based on electrostatic pressure. So instead of just not wanting to be around other ions that are the same, now we take the charges into account. So positive charges will repel other positive charges, and negative charges repel other negative charges. And negatives will attract positives and vice versa. So they would like to move so that the net charge across the membrane is zero. They want to have an equal number of positives and negatives on each side. So that's that electrostatic pressure they'd like to exchange so that everything equalizes across the membrane. So this is what they would like to do. This is why when we force that cell membrane to sit with a difference in charges across it, we're storing energy. We're storing potential energy because the ions want to move like this but can't. And so again, you might be thinking, okay, well, if the cell is at rest, how is it holding these things where they don't want to be? How did we set up this situation where there's a difference in charges? How did we force it to be in this not stable state? And this is where we introduce the last of our big players. This is the sodium potassium pump. So unlike our channels, those ion channels that allow things to sort of flow through in the direction that they want when they're opened, the pump is an active force. So from the name, sodium and potassium pump, we can assume that it's going to move both sodium and potassium. And the act of pumping things across the membrane is an active process. So it's going to use up energy. So these pumps, they're also made out of proteins. They also span across that the phospholipid bilayer. They span across our membrane, but they're going to move things using power from the cell, and they're going to set up that disequilibrium in charges. And so the way that this works, if you think of like a bicycle pump, you go up and down. And when you go up, you're releasing something outside. And then when you go down, you're pushing the air through into your tire on the downside. So when these pumps go, oops, wrong way again, when they go up, they're pushing our sodium ions, the red Na pluses, out of the cell. So they're going to hold and release 3 sodium ions outside of the cell. And then when they go in the other direction, when they pump back down, they're going to pull in 2 potassium ions. Now, we're talking about 2 different positive charges here. But because they're not equally exchanging, because we have 3 going out and only 2 coming back in, the net difference is that outside gets 1 molecule more positive, and the inside comparatively is 1 molecule more negative. So we're ending up with a net negative on the inside of the cell. So comparing our outside to our inside, we're ending up with that negative comparatively on the inside um, as we compare it to the outside. So that's how we're setting up that difference across that membrane, and that's how we're storing energy. So those sodium ions would like to come back in, those potassium ions would like to go back out, and we would like the charge to equal out, but we can't because that phospholipid bilayer is holding everything where it is now. And so we're stuck at this stage where things want to move but can't. That's that stored energy, that's that potential across the membrane. And then we can release it and harvest it to do stuff in sort of the next stages. We can talk again very briefly about our ion channels. I think we've already covered all this info, but we basically said that the ion channels are those gated pores, gated meaning that we can open and close them. They cross the membrane, they're made out of proteins, and they limit the flow of ions in and out of the cell, but we can open them if we have the right trigger. So if they are chemically gated, then we can use neurotransmitters or hormones to turn them on to open them up. Or we can use electrical charges, so changes in that electrical potential can trigger them to open. And so that's sort of our baseline. We've set everything up. We've established how we end up with that difference in charge. And I'm actually going to stop us there because I would like to review that stuff at the beginning of next class and then talk about what happens when we open those channels, how that creates signals that can then move and give us information downstream. So again, if this hasn't all completely sunk in yet, that is totally fine. We're going to do this one more time. I will also eventually post some videos on our Moodle because I do find sometimes seeing these things moving and animated helps. So we'll go over this a couple more times before you're expected to know. Yeah, we'll leave it there and we'll come back and talk about action potential.